Good morning, everybody. Welcome to the RCN webinar. We're going to wait another minute just to let everybody trickle into the webinar and we'll get started. Okay, I'm going to explain how the webinar works. So welcome everybody to the second in our new series for RCN, for our Research Coordination Network on wastewater-based epidemiology for infectious disease targets. Today we have two speakers, and the first speaker is Peter Arts, and the second speaker is Amy Pruden. And each speaker will speak for 20 minutes, and then we'll have 10 minutes of questions. And in this webinar format, everybody should put their questions in the Q&A, and you have the opportunity to upvote questions if there's questions that you're really interested in having asked by the moderators. So I have up on the screen the speakers for today, and then a plug for the next webinar, which is December 2nd, where we'll hear about RSV and also Candida auris. Okay, so with that, I'm going to stop sharing my screen and let Peter share his screen, and he can go ahead and get started. Oh, and Heseth, did you want to say something real quick?
Yeah, Kyle just dropped the link for the listserv, but keep an eye out for our upcoming workshop. Our first workshop of the series will be December 12th, but we'll be sending a registration link and more information shortly.
All right. Thanks everybody for having me here. I'm excited to present some of my data. I switched my title. My old title was a little bit of a mouthful, so I think this is a little easier to get the gist. But I've been looking at fecal shedding measurements for the application of wastewater-based epidemiology. There's a list of some of our collaborators from a lot of different places. So currently trends in wastewater measurements and correlations between measurements and clinical cases or hospitalizations are the primary source of information regarding the dynamics of infectious disease outbreaks for wastewater-based epidemiology. The figure on the left shows some of these correlations. So SARS-CoV-2 N gene measured on the x-axis, and the reported daily cases per population on the y-axis. And it shows on the left an unnormalized SARS-CoV-2 measurement and the improved correlation we see with a normalized measurement. And that's especially true looking at multiple different catchments and showing that by normalizing, we're better able to compare measurements from each catchment. And on the right, we see the clear relationship again of the normalized SARS gene measurements compared with smoothed incidence. But as you can see, there's still a good amount of clustering within catchments, indicated by color here. And there's some observed sort of spread across the trend line. And so this variation is likely caused by factors such as individual system hydraulics, wastewater composition, or spatiotemporal changes in fecal shedding, though we don't yet have sort of a robust enough data of fecal shedding to evaluate what factors are contributing to these. So these trends are useful in estimating the number of cases at any given point in the pandemic, but on their own aren't enough to accurately provide other descriptive epidemiological measures, such as disease prevalence or effective reproductive number of the virus. And observing these trends still really relies on continued testing and sort of, homogeneous rate of testing that allows us to compare apples to apples across different phases of the pandemic. So this recent paper by Jana Huisman and colleagues used wastewater data coupled with estimates of fecal shedding to estimate the effective reproductive number of SARS-CoV-2 over an outbreak in Zurich in the fall of 2020. And so they used collected wastewater data along with fecal shedding, and they compared their estimate, which is shown in green and yellow on the right plot, and compared that with an estimate from clinical cases in gray. And while that correlation, seems to track pretty well, they make the point to say that more work is needed to determine both the shedding load distribution and the amount of virus shed during an infection. Other models have been built in the attempt to directly estimate the prevalence of infected individuals from wastewater. So on the left, Jeff Solar and colleagues used a mechanistic materials balance approach to estimate the fraction of individuals who are infected within a sewer shed. And you can see that they're superimposed on the confirmed daily cases. But again, while it looks like a good correlation, they make the caveat that future work is critically needed to characterize fecal shedding of SARS-CoV-2 RNA, as well as pepper mild model virus RNA, to enhance the utility of this framework. On the right, McMahon et al. Used an epidemiological approach to essentially create a standard curve comparing the amount of RNA, SARS-CoV-2 RNA in the wastewater catchment to the number of active COVID-19 cases. But again, they cite the need for better shedding data, especially surrounding the dynamics of SARS-CoV-2 infection. So there is published data examining the prevalence of SARS-CoV-2 fecal shedding, though it's not catered to use in mechanistic wastewater-based epidemiological models. Much of the data are small-scale clinical reports meant to describe manifestations of illness and thus don't prioritize information critical for application in mechanistic models. These studies often have ill-described molecular methods that can vary significantly from study to study. And additionally, quantitative measurements are often reported as CT values or gene copies per volume of extract, with little standardization of the quantity of sample processed. Another key issue here is the solids content of stool, as solid content can vary widely between individuals and within an individual over time. And because SARS-CoV-2 partitions favorably to solids, gene copy measurements relative to dry mass may be a more representative basis when accounting for inputs into a sewage system. All this being said, fecal shedding data that is quantitative, externally valid, and spans the infection is lacking and is critical for wastewater-based epidemiology applications. So there are two studies that are sort of represent the most comprehensive data out there to date. This paper by Roman Wolfel et al. Early in the pandemic looked at 9 patients and 60 samples total and measured longitudinal SARS-CoV-2 in stool. They measured it on a per gram basis, but they didn't account for percent solids or have rigorous methodology outlining their sampling and homogenization procedures. And then a more recent paper by Natarajan et al. Measured fecal shedding on a per microliter basis in 113 individuals with mild to moderate infection. Though the stool was self-sampled by the patients and the volume of sample was not recorded prior to homogenization. So again, it's difficult to directly apply this data to a wastewater modeling framework. So to fill some of these gaps in the literature, we're collaborating with the Find COVID project centered at University of California, San Francisco. They're a CDC-funded project aiming to determine the infectious period throughout different phases of the pandemic. And in addition to individuals who have tested positive for the virus, they enroll household members who have been exposed but have yet to test positive. So that gives us the ability to probe for pre-symptomatic infectivity and pre-symptomatic shedding. So in total, we've measured around 390 stool samples from 48 individuals collected relatively early in the pandemic from the San Francisco Bay Area. And so in addition to the symptom information nasal swabs, we've been quantifying the stool from these patients as well. And while a lot of these samples are from early in the pandemic, we have plans to continue analyzing samples that they're collecting. So future samples will include different variants and vaccination status as well. So our lab uses a reverse transcriptase digital droplet PCR for our wastewater analysis, and we took the same approach to measure these stool samples. Specifically, we measured precise masses of sample to homogenize for nucleic acid extraction, and we took a paired measurement of the solid content for each of these samples as well. After a PCR inhibitor removal step, we ran a multiplexed assay quantifying SARS-CoV-2 N gene as well as ORF1A, and we also measured PMMOV on the same plate. We then ran a separate qPCR assay for crassphage as well. We combined the percent solids measurements with our quantitative measurements to get a quantitative measurement on a gene copies per milligram of dry weight basis. So we used the nucleocapsid gene as our target of choice here, as it's the same target we use for the majority of our wastewater analysis. The top plot on the right shows all of our measurements of N longitudinally based on the day post-symptom onset for each individual. And open circles represent non-detects. The bottom plot is a histogram of the number of samples we had on each day post-symptom onset. So in summary, we measured 379 samples and saw a total prevalence of 50%, including an individual prevalence of 72%. So that means out of our 48 individuals, 72% had at least one positive sample. The maximum shedding value we observed was 2.8 times 10 to the 6th gene copies per milligram of dry weight. And our sampling period covered from -3 days post-symptom onset to 28 days post-symptom onset. And we want to note here that we had 4 samples that were pre-symptomatic in individuals, and 2 out of the 4 samples were positive. This plot shows the proportion of samples that were positive for SARS-CoV-2 on each day post-symptom onset, with the point size representing the number of measurements we had for that day. The proportion of individuals shedding peaked between 0 and 5 days post-symptom onset and decreased to about 20% by day 28. Looking at the individual infection trajectories, we see a highly individualized course of infection or course of shedding. Some individuals peaked shedding early, before 10 days post-symptom onset, while some peaked later in the sampling period. Others had a more sort of stochastic shedding trajectory, shedding high, going down low, and then shedding back up again. And still others did not provide a sample that tested positive for SARS. And so we defined this sort of good threshold for sampling coverage as 3 samples with a range of at least 15 days from the first to the last sample. And we observed that 22%, or 7 out of 31, did not shed SARS-CoV-2 over this period, though it's still possible that sampling limitations prevented us from observing a shedding period here. The majority of samples we tested for PMMOV were positive, 95.9% to be exact. And every single individual had at least one sample that tested positive for PMMOV. The range of values measured ranged from not detectable up to 5 times 10 to the 8th gene copies per milligram of dry weight. So in this box plot, each box represents a different individual and all of the samples that individual had. And so what's striking to me about this is just the dynamic range of fecal shedding over such a short period of time. So this is sort of a 30-day sampling period, and people are shedding, variably, up to, 6 orders of magnitude. And so it's really, really a variable shedding measurement. Crassphage shedding exhibited a much different pattern with much less individual variability. And the sum data sort of followed a bimodal type distribution. So the majority of individuals who were shedding shed relatively low concentrations of crassphage, under 10 to the 4th copies per milligram of dry weight. while a smaller fraction of individuals shed pretty high concentrations, and an even smaller fraction shedding sort of intermediate amounts. Also, the prevalence was much lower, so around 50% of all the samples we measured were positive for crassphage, and only about 80% of individuals had at least one positive crassphage measurement. So this data can be directly applied to the models discussed earlier as sort of an improvement to the available literature of fecal shedding data. But it can additionally help us to design better and more efficient sampling infrastructure by asking some questions about what biomarkers might be useful in different situations, at what population scales are these biomarkers useful or is it not useful. Thinking about the variability we're seeing, is there some point where we're no longer improving our interpretations of the wastewater data. And then similarly to the variability of biomarkers, how does the variability we see in SARS-CoV-2 fecal shedding influence our correlations with clinical cases or hospitalizations? So in order to approach these, I took an epidemiological approach using an SEIR model, which simulates sort of an outbreak mathematically. So it's a 4-compartment model that will generate numbers of individuals in one of the 4 compartments: susceptible, exposed, infected, or recovered over time. I used parameters derived from the literature for a SARS-CoV-2 outbreak, and generated these curves of infected individuals. I then used the number of infected individuals in the fecal shedding data described earlier, paired with a materials balance derived from the literature to generate a stream of wastewater measurements paired to this outbreak. So here we see the stream of wastewater SARS-CoV-2 measurements as the gray dots, with the black line being the rolling average of wastewater solids measurements. And the red line is the infection prevalence, so the number of infected individuals over time. This was generated by defining a fecal shedding trajectory for each infected individual to follow through dynamically. So it's accounting for sort of the progress of infection of each infected individual. And this curve was generated for a population of 1,000 individuals, and sort of shows the dynamic impacts that just the biological variability of fecal shedding is having on this system. So by comparing these results with the same model run at different population sizes, we can see that the effect of individual variability is waning at higher populations. It's sort of being averaged out. So what I take away from this here is that we're sort of starting to lose the reliability of our trends when we go under 1,000 individuals with this model. So here I ran 10 models for each population level, and that's what each of those lines are. They're just sort of stochastic differences with each run of the model. And under 1,000, we're getting less and less consistent wastewater streams. And the same is true for the PMMOV and crassphage measurements. So sort of below 1,000 individuals, we're getting less and less uniform concentrations in the wastewater. So lastly, to answer the question of how high concentration shedding is impacting wastewater measurements, I experimented with generating streams of wastewater data from fecal shedding data sets. The top plot is a histogram of modeled values of fecal shedding derived from my data. And the colored bars represent the 90th, 80th, and 70th percentiles of shedding values. I ran my model 4 different times by including each data set. So starting with the entire data set, then running it again with a data set only below 90% confidence or 90th percentile shedding values, and then below 80, and then below 70. And the results are shown in the bottom left. So the black line represents the whole data set, and the colored lines represent each subsequent decrease. And so we can see here that with removing the top 10% of shedding events, we're decreasing our wastewater measurements by 50%. So the highest shedding events are having an outsized impact on our wastewater measurements, quantified here. So in conclusion, it's important to have high quality and externally valid fecal shedding data for optimal results from mechanistic descriptions of wastewater epidemiology. And we think that the dynamics of individual shedding likely influences the variability at the level of sampling and should be included in wastewater models. Some limitations, like I said earlier, there's likely differences in fecal shedding with variants in vaccination, and we hope to sort of probe that with future samples. This study was also not powerful enough to observe differences in demographics with shedding, which likely has profound implications with looking at different wastewater catchments. And we also we ran these models sort of as a simple proof of concept, but we hope that the data can be used in sort of future models to answer some of these evasive questions in WBE. So just some acknowledgements. This work was done at the U of M with some collaborators in epidemiology. The samples were provided by UCSF, which is a CDC-funded study. And this was done with funds from CDC Foundation. So thanks.
Thank you. That was lovely and a huge gap. I'm sure everybody's going to be digging into this this publication. So we have a lot of questions in the Q&A box. Don't forget to upvote questions you want to see answered. And I'm going to start rolling through them. So you talked at the beginning of the presentation about some of the limitations in the literature with respect to fecal shedding studies. Can you talk a little bit about what are the recommendations that you would provide, and if you've made those recommendations to others?
Yeah, I mean, I think it's just really important to collect as much data about the sample as possible. Different groups measure, wastewater solids versus the influent. So I don't think it necessarily needs to be on a per mass basis, but I think having that information provides you to standardize across studies. So I think that's the most important piece is, having, the density of the stool, the solid fraction, the volume, all of that is really important.
Yeah. Did you have any information about the level of disease severity in the population of people providing the samples?
Yeah, so all individuals were excluded from the study if they were hospitalized. So these are sort of subclinical, and some of them were asymptomatic. I didn't make clear which ones were here, but every individual here had at least one respiratory positive by PCR test, but some were asymptomatic as well.
Okay, I think that might have been a question down. So did you have any, did you look into any different mutations? I'm not sure when the study was conducted or when you guys collected the samples, but did you see any trends with different variants and different shedding in between individuals with BA5 versus BA4?
Yeah, so these samples were collected sort of late 2020, early 2021. So a while ago. We're sort of limited to the early pandemic variants with this cohort, but we've continued to get samples from the FIND COVID team, and we're planning on analyzing them. So hopefully get us into Delta and Omicron, and in the future.
Cool. Okay, so someone asked, instead of considering concentration, have you taken a look at the total mass of viral particles released per person to take into account variable fecal mass developed between adults or children, and along with changes in food consumption while ill? So like a mass load instead of or a copy number load, I guess it would be.
Yeah, that's really interesting. We don't have precise measurements for the mass or volume of stool produced by each of these patients. We do have age and sex and ethnicity demographics, so we could look into that, sort of stratify fecal production by age and demographic maybe, but yeah, that's really interesting question.
Okay. And then, so you said you got some asymptomatic, but I think you mentioned, were there any differences in the shedding characteristics between the asymptomatic people, or maybe, I guess this was probably pre-vaccination, but the vaccination status, let's just got uploaded.
Yeah, so we didn't have enough samples from asymptomatic people to be certain, but the asymptomatic samples we did have, they did shed fecally, and same with vaccination. We didn't have enough vaccinated samples yet to sort of determine that. But
all right. So I think you kind of got at this with your model, right? When you're looking at if you took out the top 10 percentile, but I guess the question is, how much more virus do super shedders contribute compared with an average infected person?
Yeah.
Like it has an impact, like what's that delta, I guess?
Yeah. I think that it's something that is definitely really interesting. So the model that I did was just looking at the sort of individual shedding event. So it would be interesting to look at, the individual trajectories and see which individuals are hitting those high peaks and looking at their total sort of curve. So next steps, I think, but definitely the high shedding events are contributing a lot disproportionately, I think.
And then there's questions about methods. So concentration of the solid, of the stool, I guess, extraction and inhibitor removal, and your limits of detection.
So yeah, we to sort of minimize the inhibition, we did a pretty dilute homogenization. I think it was around 20 milligrams per mil of homogenization buffer. And we use a Zymo inhibition removal kit. So just sort of a plate spin through column to take out some of the organic matter in those samples. We still did see a little bit of effective inhibition by looking, seeing sort of nonlinear decrease in signal from tenfold dilutions. But we did our best to minimize it. At most, it was, a couple fold difference, but and then our limit of detection it changed from plate to plate because there's differences in solid concentration, but it was about 50 gene copies per milligram of dry weight.
Okay. And then I'm going to ask a question for the panelists. How do you think repeated infections might impact the shedding? Did you try to match PMMOV or crossphage shedding with wastewater measures? If PMMOV and cross are typically similar concentrations, so I guess in the wastewater, we see that they're typically around similar concentrations, but in this data set, it doesn't seem to be. Do you have any ideas of why that might be?
Yeah, I think it's probably because the concentrations in wastewater are driven by the highest shedders, and sort of the maximum shedding levels that we saw were pretty similar, around 10 to the 8th gene copies per milligram of dry weight. So I think it just, the individuals that are shedding down at 10 to the 3rd, for crossphage are just not sort of changing the wastewater concentration. So I think that's probably what's happening there.
And what about repeated infections? Do you have any ideas of how that might impact things?
I mean, I'd imagine that sort of, prior immunity to SARS-CoV-2 might lessen the shedding rate in stool, but all of the individuals from this cohort were first-time infections, so it'd be interesting to look at. Yeah.
All right. Well, I don't think we got through all the questions, but I think it's time to hand it off. Thank you so much for your presentation and for filling this gap. And I'm sure there's some questions in the chat. Maybe you could answer those. Yeah.
Sure.
Thanks.
Peter, you can stop sharing your screen. Okay, Kyle, I'm handing off to you.
Okay. So thank you, Peter. Great presentation. And it's our pleasure to welcome next Dr. Amy Pruden, who's a University Distinguished Professor at Virginia Tech and a world-renowned expert in antibiotic-resistant gene fate in the environment, which I believe she's now working to translate to understand the prevalence of antibiotic resistance genes in communities using wastewater monitoring. So just a reminder to put any questions you have in the Q&A box, and take it away, Amy.
Yeah, thank you so much for having me. I'm so glad that this group has restarted because there's so much work to be done in the wastewater-based epidemiology space. And so today I'll take us in a little bit different direction. We've also been mucking around in sewage, as you can see. I've been working with Peter Vickersland, and here's two of our students, Ben Davis and Matt Blair. But we've been taking it in a bit different direction, looking at antimicrobial resistance. So I'm very sorry to be the bearer of bad news that we're in the midst of another pandemic. It's being called the slow pandemic. I don't know if that's good or bad, but antimicrobial resistance is basically where our drugs, including antibiotics and other antimicrobials, antifungals, et cetera, no longer are effective for curing deadly infections. And so it's estimated that in the US and UK, there's about 35,000, 25,000 deaths per year. In India, close to 60,000 deaths per year just of newborn babies alone. And it is difficult to get firm numbers on this slow pandemic, but this paper came out in the Lancet just this year, but tracking 2019. And, this was a very comprehensive study where they looked at 23 pathogens, 88 pathogen drug combinations across 204 countries and territories. And so they estimated that there were about 4.95 million deaths per year globally, what they called associated with bacterial AMR specifically, so antibiotic resistance. And so you can compare that with COVID, so it's pretty substantial. So in 2019, my collaborator Li-Ching Zhang in computer science approached me. We'd been working for some time looking at antibiotic resistance genes in sewage with my collaborator Peter Vickersland. And she said, hey, we should think about the wastewater treatment plant of the future, and we can equip it with cyber infrastructure for monitoring antibiotic resistance genes. And I thought, wow, that's a really cool idea, Li-Ching, but kind of out there. I don't know if the world is ready for that. But as we all know, we put in this grant 2019, the world changed in 2020, and the world apparently was ready for this. And so this project started in 2020. And so that's what we've been doing, basically working towards building cyber infrastructure to monitor antibiotic resistance genes in wastewater treatment plants. But it's important to point out that antimicrobial resistance is not COVID-19. And so, for example, the Lancet study that I pointed out, there were 23 bacterial pathogens, 88 bug drug combinations, not to mention that there's over 3,500 ARGs curated to date. So as complicated as SARS-CoV-2 monitoring is, this is a bit more complicated. So especially in terms of what do we even monitor and where do we monitor? So it's worth spending a little bit of time talking about what exactly is AMR, or specifically antibiotic resistance. So focusing on the bacteria, so the ability of bacteria to survive antibiotic treatment, or inability of the antibiotics to cure the infection. So at a mechanistic level, if this is a cell, then it could be pumping the drug out, it could be modifying the target, it could be degrading the antibiotic. This is all encoded in antibiotic resistance genes. Now, how do we observe that? Of course, we could use DNA-based methods, but we can also plate the bacteria. And then if we put discs infused with antibiotics, the resistant ones will be unfazed by that and grow right up next to the disc. Now, a complicating issue is horizontal gene transfer, and that's because bacteria can actually share genes with each other. And so that means that if we're tracking antibiotic resistance, it actually makes sense to track the genes and not just the pathogens. And we're looking in sewage for the same reason folks have been looking at SARS-CoV-2, right? So when folks ingest antibiotics, they select for a resistant microbiome, and that's going to end up at the wastewater treatment plant. But in this case, we are interested in what comes out the other end of the wastewater treatment plant, because it is possible that resistant bacteria and the ARGs can end up in the environment and could still pose some risk in terms of spreading antibiotic resistance. And so we did take the opportunity to publish this current opinion piece in 2021, kind of laying out the opportunities that now that we've invested in this infrastructure for sewage surveillance, that there's a lot of opportunity to look at antibiotic resistance and other public health concerns as well. So around the same time in 2019, we put together a proposal to the Water Research Foundation, and they were proactively making the call, like, okay, we think AMR is a concern, tell us what to monitor. And so this was also awarded in 2020 and had teamed with Jody Harwood at the University of South Florida and Lenny Heath, who's in computer science, and had several amazing students working on this project. And so basically trying to see, okay, you know, AMR, there's so many targets, it's a moving targets. What would we even suggest that the water industry could monitor? And part of this was getting expert input. And so we did an online survey globally, and we also had a workshop. And this was really nice to be able to do this on Zoom, that we could get lots of international participation, others working on similar endeavors, as well as a lot of representation from some of the federal agencies. So CDC, USDA, FDA, EPA are also working on standardizing methods for monitoring AMR in the environment. And also got water utility perspectives to try to rein this in and make sure that what we're proposing is feasible. And we recently published this framework paper in ES&T led by our student Krista Luguari. And in a nutshell, basically, we quickly realized that there is no one target that we should be looking at in terms of AMR. And so where we landed was that it basically depends on your monitoring objective. And we identified 4 monitoring objectives. And the first one is most relevant to this group. So basically monitoring resistant bacteria and ARGs that are circulating in human populations. But again, we're also interested in, for example, what might be evading treatment, what are the removal efficiencies of different treatment processes. And then unique to AMR is we really worry about the evolution of a new superbug. So are there certain conditions that are conducive to mutation, horizontal gene transfer, and selection of new resistant strains? In terms of targets, we kind of broke it down in terms of culture, qPCR, and metagenomics. And culture-based monitoring has a lot of value. This is your classic microbiology, and it's really the only way to know that you have a viable pathogen. And you can profile it, you can assay phenotypic and genotypic resistance, you can do whole genome sequencing, which is really useful for source tracking. And it's very useful if you're doing human health risk assessment. So a lot of folks were behind E. coli as a logical starting place, especially ESBL E. coli. The World Health Organization recently developed a standard method for that could be built off of. Enterococci is also attractive because then you have a gram-positive organism, and there's different antibiotics that we're concerned about with those organisms, and also have really nice standard methods for wastewater. But there were others that were looked at as well, and especially, you know, we should think about our fecal indicators, the best option or only option, or maybe we can look at things like aeromonas and pseudomonas that actually persist and grow more in the environment and have the opportunity to engage in horizontal gene transfer. qPCR is nice because you avoid culture bias, right? It's hard with culture just picking one organism or the other. With qPCR, we quantify that ARG across the whole microbial community. And it's also nice as a quantitative measure to assess removal rates and to inform risk assessment and other models. In terms of what to monitor, this Class 1 integrase gene in Taiwan is pretty popular, as is SUL1. They're very strong anthropogenic indicators. So you measure them at nice high levels, and then you can see their attenuation and removal. But there's also interest in some of the genes that encode resistance to last resort antibiotics, right? So some of these BLA genes encode resistance to extended spectrum beta lactamases. But still, you're kind of stuck picking which genes to look at. There is a high throughput qPCR approach that can help with that. And then finally, the third category of methods we looked at was shotgun metagenomic sequencing. So in theory, this is the answer to all our prayers that, you know, one method, you wave a wand and you get sequences of all the genes in the sample. But for any of you who have actually done shotgun metagenomic sequencing, unfortunately, it doesn't quite work that way. And you do have some limitations, especially high detection limit, because you're sifting through a lot of non-target DNA, and it is expensive and it requires a significant level of expertise. And it's kind of the Wild West in terms of data analysis approaches that have to be worked out. But there's a lot of excitement that there's potential here. And even amongst the water utility participants at the workshop, you know, they were kind of looking to this as a method of the future. And especially when it came to wastewater-based surveillance, I think they were excited about that possibility. So I want to spend a little time talking about metagenomic-based methods. And this is a paper that came out recently that was part of a partnership in international research and education grant through the NSF that Peter Vickersland was the PI on. And we got to send students around the world to sample wastewater treatment plants and do metagenomics and to annotate the antibiotic resistance genes. So you see these colors here correspond to various classes of antibiotics that you might recognize. And when we ranked from high to low, we saw a pretty striking pattern that's highest in Hong Kong and India and lowest in Sweden. And so this really stood out to us because if you follow antibiotic resistance policy at all, Sweden is really well known for being one of the most proactive, if not the most proactive country on earth in terms of aggressive policy to combat the spread of antibiotic resistance. And so this really opens the doors. It's exciting that this kind of monitoring data could help to inform and evaluate policy. That was all short read sequencing data. So long read metagenomic sequencing data is nice because when you get a nice long read of DNA, you not only see the ARG, but you can see adjacent DNA. Is it on a plasmid or another mobile genetic element? Which bacterium is carrying that ARG? And so this is a study of the same samples, just zooming in on one of the wastewater treatment plants. So Dongjuan Dai did an in-depth analysis of influent sewage compared to activated sludge. And kind of what jumps out in this network analysis is that the associations between ARGs and putative pathogen annotations really reduce in the activated sludge. So that is encouraging that the activated sludge is a good ecological barrier to ARGs, but there are still ones that do escape. And so those are ones we might want to be concerned about. And in this study, this was a study led by our student Hania, where she did 18-month longitudinal monitoring of a local wastewater treatment plant. And one of the interesting things she saw, these core diagrams are basically matching up what we call bug-drug combinations, so pathogens and resistance classes. What really jumped out here is that the effluent bug-drug combinations really showed a more similar pattern to our regional antibiogram. So that's like people who present themselves in the clinic with infections and they actually isolate the bacterium and do the profiling. So that was a better match with effluent than it was with influent. And so to us, it really emphasized that it's valuable from an AMR standpoint to also monitor the effluent, not just because it gives us removal rates, but it's also potentially telling us something about clinical resistance. And so now I'll share a little bit of our preliminary data so far from the sewers project. And so at this point, we have over a year of data sampling twice a week, influent and effluent, at a local wastewater treatment plant. So it's a neighboring town to where Virginia Tech is. And then here in Blacksburg, we've been comparing. And so in this map, it's just showing that different counties in Virginia have different socioeconomics in terms of, you know, we have unemployment rates and populations. So we also got a few samples from Northern Virginia to mix that up. We annotated ARGs from the CARD database and over 100 pathogens. And so Connor Brown has been leading up a team of students doing this analysis. And so here's some interesting data. So lots of annotations of ARGs and the influent and effluent. And one of the things we're interested in are potential anomalies. So this kind of jumps out here, right? So we'd say, uh-oh, here's streptogrammin resistance, and that is a clinically important antibiotic. So, you know, what do? Do we sound the alarms? Well, we look closer at the data. And so we saw that when we assembled the data, that it wasn't really necessarily a clinically relevant form of resistance. It's in these cyanobacteria. So there was an algal bloom basically, and that could be related to an operational problem with the plant. So they had an F to M ratio problem. And this is just starting to play around with the data a bit more. So one of the last resort antibiotics we're really worried about is colistin resistance. And so this is a family of genes, so MCR, and these are in the same family, just so it's showing that, yes, we're finding them in the influent. We're also finding them in effluent. They have different genetic contexts in the Burkholderia and the Aeromonas. And so this is something we want to look at a bit closer. And then we've been working to annotate an array of bacterial pathogens, so you can see those here. And again, we have to remind ourselves that we don't have the resolution to say for sure it's a pathogen. So they're putative pathogens, but it shows us some trends that could then be followed up with. And so here we're looking at Blacksburg, neighboring town of Christiansburg, and then we have a little more sparse data in Northern Virginia. But it's just interesting to compare some of these patterns. And a lot of them relate to sewage temperature, which I guess is not a big surprise. So if we're encouraging everybody to collect these kind of data, we need to encourage consistent format and sharing. So that's what we're trying to do with the water antibiotic resistance database. And this is being led by my co-PI, Lenny Heath. And this is publicly available. Feel free to check it out. We're looking for beta testers. Basically consists of Excel spreadsheets to make sure people report data consistently and upload it. And then we can see large scale trends and start to support some broad modeling of the data. So in summary, in our current opinion paper, we lay out a lot of advantages to monitoring AMR in the environment. So that's wastewater treatment plants, but also surface water. So there's several here that we've indicated. And just that, again, there's a real opportunity here. So, we've invested in this infrastructure for wastewater-based surveillance. And I think this group's probably aware that the CDC recently put out a call like, okay, let's expand on this for multiple pathogens. And that includes ARGs. And so we were recently notified that we will be the recipient of one of these grants. And so we will be very active in this group, trying to learn from everybody here and move this forward. And finally, just wanted to point out that ES&T just had a special issue released that's focused on antimicrobial resistance in the environment that I hope will be a handy reference for you all. And with that, I thank you for your attention and point out we are recruiting PhD students. We have a NSF NRT focused on combating antibiotic resistance. And so we look forward to continuing to move this forward. And I look forward to any questions that you all might have.
Great. Thank you. Very nice talk, Amy. Just a reminder for folks, if you do have questions, to put them in the Q&A box and upvote the questions that you're interested in. So the first question we have is from Rose Cantor, and there's a couple of upvotes and sort of similar questions, but basically, have you explored any enrichment approaches for your metagenome sequencing to better target ARGs, so probe-based or other enrichment approaches?
Yeah, that is a very insightful question. And so there's kind of this two-edged sword. So we have this line of research to assess the extent to which metagenomics is quantitative. So, some skeptics will say, oh, metagenomics is not quantitative. Well, we're showing that it is within a certain range, and that's really valuable for, if you're assessing removal rates or doing any sort of modeling. But yes, the detection limit is there, and that's an issue since it's a non-target analysis. And so I think the reality is the enrichment makes sense, especially if your goal is specific pathogens that you're interested in and you have a short list, specific pathogens, specific ARGs, and you just want to know, are they there? And you're not as worried about the quantity, but it could get you straight to what you're interested in. And then you can look at the sequences and see if there's variance. So yeah, I think that makes a lot of sense.
And somewhat similarly, do you remove human DNA or any other signal from your metagenome? I guess either before or after sequencing.
Yeah, I mean that all gets into your normalization methods. And so yeah, we pretty routinely search for human DNA and pull that out of the analysis. And we have actually a paper in review right now where we look at all different ways of normalizing the metagenomes, because that's another part of the Wild West. Some normalize to the 16S gene, some to the RPOB, some do RPKM. And so we did kind of a meta-analysis of these different normalization methods. But yeah, that's human DNA is something you have to watch out for.
Great. So I guess changing gears a little bit, do you know which ARGs the CDC, or I guess you all as funded by the CDC are specifically targeting?
Oh, maybe move on to the next question and I'll pull that up because I'm trying to remember. I
Okay.
know the
All
BLA,
right.
I think it's BLA CTXM, which is a carbapenemase. Yeah, I'll look up the other one while you ask me the next question.
Okay. So can you provide any more details about this ward database and what sort of things you're looking for with beta testers?
Yeah, so I should point out it takes metagenomic data, but also culture-based and qPCR-based. So anybody who's doing any sort of monitoring of either ARGs or resistant bacteria in the environment, you know, they're welcome to try it out. And the ARGs are BLA CTXM, BLA OXA1, and then MCR1, which is the colistin resistance gene that I was talking about there at the end. So that's what we've chosen to focus on in our CDC project.
Great. So another question is what do you think of the use of antibody grams, or sorry if I mispronounced that, for wastewater like it is used in hospitals?
Well, I don't know. I mean, what I shared was just observational. But since that is the gold standard of hospitals, you know, first of all, we need more clinical surveillance, right? That when people get infections, you know, encourage your doctor to actually culture it and profile it, because then you know better which antibiotic to use, right? But doing the same in wastewater, and then we can start to see the degree to which they match up.
Great. And I think we might have time only for one or two more questions, but from Preston Mansfeld. What do you think about the impact of the sewage systems in general, the conveyance systems and the population and population size? So he pointed out that Hong Kong is heavily invested in separating their system. Stockholm is maybe largely combined system. You know, how do you think that drives observations beyond maybe sort of a true antibiotic resistance prevalence in those associated populations?
Yes, very insightful question. So, I mean, the bottom line is, to what extent is sewage representative of human fecal material? And the reality is the sewage conveyance system is its own ecosystem, right? So you've got biofilms and all sorts of things happening from point A to point B, not to mention infiltration and everything else. Hong Kong, I'd also point out, they flush their toilets with seawater in a lot of cases. So then there could be some saline stress that's happening there. So we have another study and review that we're comparing sewage and fecal resistomes. And so, you know, hopefully that'll be out soon and I can tell you more. But I think kind of the upshot is the sewage in a weird way told us a lot more than the human fecal resistomes. I think because at the end of the day, the fact that it is this composite sample, the trends were a lot stronger for what we were looking for than when you're trying to look at individual human feces samples.
Great. Well, I think we're out of time. So just to keep us on time, we weren't able to get through all the questions, but thank you to both speakers for excellent talks. And I'm sure if the folks wanted to continue the conversation, they would be open to that. Thank you everyone for attending. Just a reminder, if you haven't signed up for our listserv, to keep up to date. And our next webinar will be Friday, December 2nd at this same time. So thank you everyone and have a great weekend.